to talk about the HIPO-YAP in fibroblast activation and kidney fibrosis. Let's get started. As you may know that chronic kidney disease is a growing public health problem worldwide. More than 200 million people have chronic kidney disease. In the United States, in 2014, about 13% of the population have chronic kidney disease. 2020, so it's more than 14%, and it's estimated this prevalence of CKD in the US is going to be near 17%. So this is a huge public health problem. And the cost for taking care of this patient are quite high. This is a USRDS report 2018 can show that from 1996 to 2016. This orange line is the expenditure in billions for taking care of a patient with chronic kidney disease. You can see that it's more than $50 billion spent in 2016. It's almost 20% of this total Medicare costs. So it's a significant expensive disease. As you may know, this renin-angiotensin-aldol system really play an important role in the pathogenesis of chronic kidney disease. See this angiotensinogen produced by the liver, then converted by renin, which produced by the kidney, produce angiotensin 1. Angiotensin 1 converted by this angiotensin converting enzyme produced by the lung produce angiotensin 2. Angiotensin 2 is a very potent vasoconstrictor, can lead into hypertension through its action on these AT1 receptors, which can also cause increased production of aldosterone by the adrenal gland, which causes salt and water retention, and also can also produce oxidative stress and leading to proteinuria, and the patient develop hypertension and chronic kidney disease. Okay, there was a trial to show whether inhibition of this signaling pathway can improve the outcomes of patients with nephropathy or kidney disease along with diabetes, because diabetes is really a major cause of chronic kidney disease. It's called a renal trial. It's about 20 years ago, this trial. It's a double-blind, randomized, placebo-controlled study. I can show this results show that this medication called losartan is a angiotensin 1 receptor blocker. They call ARBs. Really, compared to placebo, is leading to 18% reduction of decline of the kidney function. So that's this is very, very good. You know, that's establish the cornerstone, use this medication for treatment of diabetic kidney disease. Okay, so this next is come to the on-target trial to see, you know, whether one medication is better than others. So telmisartan, similar to losartan, is angiotensin 2 receptor 1 blocker, and rampril is an ACE inhibitor. Can see these two medications has a similar result in terms of the primary outcomes, or this a hope other outcome, cardiovascular death, MI, or stroke, very similarly effective. And the combination, they want to see whether this combination of these two medication is better than one medication. Disappointingly, combination is really not additive effect, so similar. So suggests that one medication is enough for this inhibition of renin-angiotensin aldosterone system. And furthermore, this study showed that if a combination of these two medications can have a serious side effect. So 10 years later, there was a trial. There's 20 years, 10 years later, there's another advancement in the treatment of chronic kidney disease. It's called sodium glucose cotransporter inhibitor, SGLT2 inhibitor. Because as you know this, you might know this is the units of the kidney is called nephron. Nephron has a filter units called glomerulus, and also tubule responsible for absorption, secretion of absorption of good stuff, secretion of waste product. So glucose is a small molecule that can be freely filtered by this glomerulus. Okay, then most of them completely absorbed, about 90% through this sodium glucose cotransport 2 in the proximal tubule, and 10% absorbed by sodium glucose cotransport 1. So recently, there's a development this SGLT2 inhibitor, originally used to treat diabetes, because diabetes have high blood sugar. If you block absorption of this glucose, so the glucose will be secreted by the kidney through the urine. So this can lead to a reduction of blood glucose, and also this glucose can act as osmotic diuretics, because glucose is osmotic molecules which generate osmotic pressure along the tubule. That's the cause of water excretion. So therefore the net effect shown here, glucose in the urine, loss of calories, and also reduction of blood pressure. So recently, there are several trials show that this class of medication are very good. Initially, they try for diabetes, and then diabetic kidney disease. And this trial recently published in the New England Journal of Medicine 2020 showed this dapagliflozin, which is an inhibitor of sodium glucose cotransport 2. So they want to know whether this medication compared with the placebo reduce the risk of renal and cardiovascular outcomes in patients with chronic kidney disease with or without diabetes. Okay, so this GFR, as you might know, GFR in the healthy person is 100 to 120. When it's low, that means you have disease. Okay, so this GFR range is 25 to 75, and also they have albumin in the urine. Usually, albumin is not present in the urine. When it's present, that means the filter, glomerulus, is leaking protein. That's not a good thing. So that's this patient also has a protein albumin in the urine, about 200 to 5,000 milligram per gram of creatinine. So that's a ratio estimate how much albumin in the urine. I can see this, they did a randomized trial. So one group received a placebo, the other group received this medication, is 10 milligram once daily. And they follow up for 2.4 years. Uh, and that's the results. This primary outcome is sustained, equal or at least 50% EGFR decline or end-stage kidney disease, meaning this patient require dialysis or kidney transplant, or death either from the kidney or cardiovascular disease. Okay, so this primary event, this follow-up over months. Then you can see compared to placebo, this medication dapagliflozin cause nearly 40% reduction of these primary outcomes. And there's also for the secondary outcomes, you can see there's also significant reduction of all-cause mortality. That's more than 30% compared with the placebo. So that's it's really exciting. First trial, losartan 2000, and then 20 years later, we have a new medication to treat chronic kidney disease. So the conclusion of this study is in patients with chronic kidney disease and without type 2 diabetes. This medication significantly reduces the risk of kidney failure and reduce the risk of cardiovascular death, heart failure, hospitalization, and more importantly, prolonged survival. That's very impressive. So that's this seems the beginning of a new era in kidney protection we can offer to our patient with chronic kidney disease. Before we usually see patient, oh, what can we do when the patient have chronic kidney disease? We always say, diet modification, do not have too much protein, fat, exercise, blood pressure, and blood sugar. But now we really have new medication we offer to our patient. But remember, there's only 30-40% reduction. It's because we need to do better, with this wide spread of this chronic kidney disease. How can we do better, for our patient with chronic kidney disease? And so the drivers in the progression of chronic kidney disease, hemodynamic dysregulation, we have ACE inhibit, ARBs, metabolic disturbance, we have this sodium glucose co-transport 2 inhibitor. But the other important factor of chronic kidney disease, is inflammation and fibrosis. Inflammation is a accumulation, infiltration of circulating inflammatory cells into the kidney, leading to this cytokine and chemokine production, oxidative stress, eventually fibrosis. So fibrosis is a hallmark of chronic kidney disease, so which is defined by excessive production, accumulation of extracellular matrix, which can lead to progressive loss of kidney function. Currently, there is no effective therapy. So but this is heavy focus on this fibrotic disorders because fibrosis is a common pathway leading to a organ failure. You can see there's a excellent review article published several years ago in the New England Journal of Medicine show that there's a fibrosis is not unique to the kidneys. There's a fibrosis of the heart. Cardiac fibrosis can lead into heart dysfunction, heart failure, and also arrhythmia and sudden cardiac death. And there's also pulmonary fibrosis. Fibrosis of the lungs can restrict lung function, cause a hypertension of the pulmonary system, and also heart failure of the right side heart failure. There's a liver. Liver is not an exception, and I see many patients in the hospital, with cirrhosis. They call it cirrhosis in the liver. This patient, liver is a very important organ. Is detoxing, metabolism. A lot of things the liver do. If they have cirrhosis, they are not performing their function. They have this can have this hypertension, ascites, bleeding, and also affect the kidney. They call it hepatorenal syndrome, and eventually they might develop cancer. Affect their brain function. Because it detox too many toxins, leading to this hepatic encephalopathy. Of course, pancreas fibrosis, patient has diabetes and or alcoholic. They have pancreatic fibrosis that they cause pain and also cancer. Skin, also there's scleroderma. That's a very devastating disease. And also there's a patient that receive this contrast, special contrast. They can have nephrogenic systemic fibrosis because the kidney is not functioning well, and you give this contrast for MRI, and they cause fibrosis of the skin. Very devastating disease, and it's usually irreversible. So now it's better because we recognize this disease, and we try to avoid to use this contrast when patient has a declined kidney function. And also there's an improvement in the contrast. There's not much osmo in this contrast, gadolinium. So that's the gadolinium based contrast agent. So in term, what's leading to fibrosis? So this is really complex, disorder. So a number of pathways have been proposed, for the fibrosis, like TGF beta SMAD signaling, integrin, also linked kinase, and growth factor, cytokine, vasoactive peptide like angiotensin 2, all can activate this MAP kinase, ROC, and JAK-STAT signaling pathway. That's leading to expression of extracellular matrix protein, leading to this fibrosis of the organs involved. So in the past, really has been focused this TGF beta 1 SMAD signaling, particularly SMAD3. So that's TGF beta 1 has shown to be the most potent pro-fibrotic cytokines. It's binding to the receptor 2, then activate this receptor 1, leading to SMAD3, SMAD2 phosphorylation. They form dimers, heterodimer with SMAD4, translocate to the nucleus, then leading to this activation of fibrotic genes and resulting in fibrosis. When I first become the faculty, we studied SMAD3 signaling in bone marrow derived fibroblast activation during the development of kidney fibrosis. But this signaling is not that straightforward. That's has you say that's like TGF beta, SMAD, then translocation to the nucleus, but also controlled by other regulatory factors. This can regulate this SMAD3 phosphorylation, and also they have activators, co-repressors, so regulate SMAD DNA binding and gene regulation. Okay, so when we started, looking into this, we decided to see what's regulate this alpha smooth muscle actin expression, because as you may know that alpha small is really a marker of myofibroblasts, which called activated fibroblasts, a major cells that are responsible for this extracellular matrix production. So we designed this this promoter region, alpha small promoter DNA. Then we include this coupled with evidence system. Okay, so we incubate this nuclear extractor treated with TGF beta 1 or vehicle. Then we use this magnetic beads coupled with the streptavidin. This actin streptavidin will combine together. We use magnet. We isolate a specific binding protein of this binding to alpha small promoter DNA. Then we did Western blot analysis, and this subject to mass spectrometry. We look into this data, so we identify some, known this TGF beta regulated proteins binding to the promoter region of alpha small muscle actin, such as SMAD2 and SMAD3. They bind into this alpha small promoter DNA after treatment with TGF beta. And this new protein called YES associated protein, short for YAP. Also in the control condition, we found this protein binding to the promoter DNA of alpha small. After treatment with the TGF beta, this is not binding to the alpha small promoter DNA, suggesting this protein might play a role in the regulation of alpha small gene expression. So what is YAP? It turns out YAP is a central player in the hippo-YAP pathway. Can see that in response to a number of signaling like LPA, sphingosine-1 phosphate, or thrombin, leading to G protein receptor couple receptor activation and rho kinase, then actin polymerization, and also cell-cell contact can leading to this merging, then phosphor this MST1-2 become phosphorylated, which subsequently phosphorylate less kinase. Less kinase phosphor YAP. YAP usually, in the is phosphorus is become inactivated. So this YAP phosphorylation of YAP is inactivation. Okay, compare normally, when you have phosphorylated protein, they become more active. After phosphorylation, they are sequestered in the cytoplasm plasma, some for degradation. And unphosphorylated YAP stay in the nucleus, regulate a transcription of this protein. Many of them related to survival, proliferation, or cell cycle regulation. And then there's a CDGF regulator of fibrosis. And also there's a negative regulator of this HIP-YAP pathways. So what does it do of this YAP? It's not really clear. Some people show that maybe YAP is a good thing. So we decide to take a look whether it's TGF-beta regulator YAP in this fibroblast. Okay, so there's a TGF-beta 1. We treated fibroblast. As you can see here, that TGF-beta 1 dose dependently upregulate of fibronectin and collagen type 1. This is two major extracellular matrix protein. And also this intracellular protein alpha small is a marker for activated fibroblast. That's basically, this confirms a published studies when this TGF-beta 1 dose dependently leading to fibroblast activation, okay, produce this extracellular matrix protein. And also, we look at this YAP and SMAD. Can see that YAP TGF-beta treatment leading to YAP phosphorylation in a dose-dependent manner. I can see that phosphorylated YAP is not active. That means the TGF-beta 1 treatment inactivate YAP. However, as consistent with the published studies, TGF-beta 1 leading to SMAD3 phosphorylation that's activate SMAD3. I see that we did further immunofluorescence staining. I see that after treatment of TGF-beta 1, SMAD3 shown in red here, so translocate from the cytosol to the nucleus. Okay. However, in contrast, YAP shown in red here in the vehicle-treated cells, it's mainly present in the nucleus. Okay, and after TGF-beta treatment, so is translocate to the cytosol. You can see here, suggesting that this YAP may play a role in regulate TGF-beta 1 induce fibroblast activation. So next we decide to see what is really YAP do using this shRNA to knock down of YAP. You can see here, this YAP is induced in response to TGF-beta 1. And then after we using shRNA to knock down YAP, and see we did this alpha small promoter luciferase activity assay. Can see in response to TGF-beta, alpha small promoter is activated after we knock down YAP. A baseline increase and further increase with the TGF-beta, suggesting that YAP is really inhibit this fibroblast activation. See, Western blot also show that we knock down YAP, fibronectin, and collagen type 1 also upregulated, indicate there's more extracellular matrix protein production, and alpha small is a marker for activated fibroblast also increase. No change in this SMAD3 phosphorylation, suggesting that does not affect. We further confirm this with the expression of this constitutively active YAP. So we transform this, transduce these cells, transduce these fibroblasts with the constitutively active YAP. You can see here, it's more expression. I can show we did the promoter assay again, show that the SMAD3, without SMAD3, this treatment of TGF-beta, there's an increase of alpha smart promoter activity. Okay, after we transduce this YAP, compare here to here. Let's focus, okay? Constitutively active YAP, as you can see, it's inhibit this activation of this fibroblast in response to TGF-beta, okay? And also inhibit SMAD3 overexpression, induce this alpha smart promoter activities. This suggests this YAP is really a repressor for fibroblast activation. And also we express this for this fiber to see whether this fibroblast, you can see that the expression of constitutively active YAP, okay, leading to this less expression of fibronectin and collagen type 1, as well as alpha small marker for fibroblasts. I see. In contrast, CTGF is induced. That's consistent with the publication. Okay, so again, this we also look at the cells, fibroblasts. I can see when the control cells, you see when look at this cells has a stress fiber formation. So that means they become more active instead of this cobblestone structure. Okay, so it says, and then knockdown expression of this constitutively active YAP show that less stress fiber formation and less activation of this fibroblasts. Okay. So next we check whether YAP affect SMAD3 phosphorylation, because as I mentioned before, phosphorylation of SMAD3 is really important for its activation, mediator of TGF-beta signaling for fibroblast activation. Can see this SMAD3 phosphorylation, no effect, either as early time point or late time point. Okay, so that's and also how about because as I mentioned before, SMAD3, translocate to the nucleus once it's activated. We wanted to know whether this activation of YAP affect this nuclear translocation of SMAD3. And this cytosol prep, nuclear prep, you can see that in the basal condition, there's no nuclear translocation in response to TGF-beta 1 and SMAD3 translocate to the nucleus. And after overexpression of this YAP 5SA constitutively active YAP, see there's a similar, suggests this does not affect this nuclear translocation. So lemon AC is a control for the nuclear loading, and GABA DH is a control for the cytosol protein. Beta actin is all present in the cytosol and the nuclear. Can see this very good controls. That means the loading is accurate and equal. All right, so next we did a chip assay to see, what does it really do, what does this YAP. Can see that with alpha smart chip PCR assay, I see that in response to TGF-beta treatment, alpha smart 3 is binding to the DNA promoter, okay, alpha smart promoter region, shown here. while, as I showed you before, that YAP is mainly binding to the alpha small promoter DNA in the rest condition. And in response to TGF beta treatment, is dissociate from alpha small promoter, suggesting this alpha small function there as a repressor to repress fibroblast activation in and basal condition and with TGF beta. So, because TED transcription factor is required for YAP signaling, because YAP has no DNA binding site. He has to partner with other transcription factors so that to regulate transcription. So TED has been shown, partner with YAP regulate gene transcription. Okay. So we test whether this YAP effect depends on TED transcription factors. So we basically transfect this mutant, YAP mutant, called YAP5SA94 alanine mutation. So this protein cannot bind to TED. Okay, so compare, this control, you treat with TGF beta, is induce this alpha small luciferase activity, and then we knock down this, show that's not knocked down this a TED binding deficient, this a plasmid expression of this YAP protein can see this potentiate, make it enhance this alpha small promoter activity, suggesting is not able to repress, like I showed you before. Here YAP5SA repress this, but if you don't have the TED binding domain or this sequence, you cannot bind into this TED that cannot function as a repressor. In fact, it's enhance this promoter activity. And we also did a knockdown of TED. As you can see here, in response to TGF beta treatment, and this is activated and knocked off TED also potentiate this alpha small promoter activity. And we did Western blot confirms this when we knocked off TED, fibronectin is expression increase as well as alpha small. This show TED knockdown successfully. Indicate that TED is also, along with the YAP, repress this fibroblast activation. Okay, so we also did this a Western blot to show whether overexpression of TED1 can have effect on TGF beta, induce a fibroblast activation. Can see here that's fibronectin, collagen, as well as alpha small. That's all induced in response to TGF beta 1 treatment compared with the controls. When we overexpress of TED1, so this is inhibited. TGF beta 1 induce this protein expression in this normal red kidney fibroblast. Okay, suggesting this TED is really function as a repressor of this fibroblast activation. Okay, so we did this. Next, we did, we want to know how come, YAP, TED, sometimes as a activator and sometimes as a repressor in cell type and context dependent manner. So we did a tandem affinity purification of YAP and this subject to mass spectrometry. Can see we identify a number of proteins in this system. Some of them are known, already identified YAP interacting protein, but this two protein, CTBP1 and CTBP2 are new, has never been identified. They call the C-terminal binding proteins. And there's two isoforms, and they might be regulation. They has been shown as a transcriptional co-repressor in this other cell system, particularly in these epithelial cells. Next, we want to know whether YAP interact with CTBP1. So we overexpress this MIC tag CTBP1 and flag YAP. Can see there's a good interaction. This IP, and that's we did the Western blot. You can see this present in here in this cell lysis. And similarly, we did also endogenous IP with YAP. I see this we able to detect CTBP interact with YAP, suggesting this might be has a role, and we then see whether CTBP1 binding to this promoter region. And see we did a promoter pull down assay. I see YAP is there and the basal condition, and also so is CTBP. I see here after treatment, they become less dissociated from this alpha smart promoter region. I see when we overexpress CTBP1, it's repress. It's really repress this activity of alpha smart promoter in response to TGF beta or and basal conditions. That's And we also knock down CTBP1. As you can see, this fibronectin alpha small expression higher than this scramble RNA treated cells. And we saw that CTBP1 knockdown and more fibroblast activation, consistent with the concept that the CTBP1 act as a co-repressor of fibroblast activation. Then we move on to see whether YAP is induced in the kidney. And we did staining. The yellow is alpha small, indicate activated fibroblasts. And the red is YAP, and the DAPI is staining for the nuclear. Can see this YAP activated in this fibroblast along with these epithelial cells. I see this is YAP is induced in the kidney, and so it's phosphorylation, meaning inactivation of YAP during the development of this fibrosis. By the way, this is a control kidney, and this UUO kidney, meaning this kidney has unilateral ureteral obstructive injury. Basically, this injury causes fibrosis. This is the most common used model of kidney fibrosis. And we generated fibroblast specific YAP knockout mice. And we show that this reduced YAP expression in the kidney and also is enhanced fibrosis. Consistent with YAP is a repressor, co-repressor of fibroblast activation. Okay, so that's to summarize. So I hope I convince you that the TGF beta causes YAP phosphorylation, cytosolic translocation. Therefore, inactivating YAP in the kidney fibroblasts. knockdown of YAP with shRNA promote TGF-beta1 induce fibroblast activation, while overexpression of YAP suppress TGF-beta1 induce fibroblast activation, indicating that YAP is a co-repressor for fibroblast activation. YAP suppresses TGF-beta1 induced fibroblast activation by inhibiting SMAD3 binding to alpha SMAD promoter region. The inhibitor effect of YAP is dependent on TED transcription factors. So TED and YAP complex recruit this CTBP1 to the promoter region of alpha SMAD, thereby repress the transcriptional activation of fibroblasts. We show fibroblast specific disruption of YAP aggravates kidney fibrosis in vitro. So to conclude, YAP-TED CTBP complex function as a transcriptional repressor of fibroblast activation through inhibition of TGF beta SMAD3 signaling pathway. So activation of YAP signaling may represent a novel therapeutic strategy for fibrotic kidney disease. I would like to thank people in Zhongmo did most of the work, other people in the lab, and also collaborators at UConn and my former institution, Baylor, and other collaborators around the states, and the funding from NIH, Department of Veterans Affairs, American Heart Association, some private foundation, DCI. Thank you very much all for your attention. Thank you.
Thank you, Dr. Wang, for your great talk. So I really enjoyed it. So I'm gonna get questions from the audience.